just going to take a moment and just summarize the different types of membrane junctions that we talked about in Chapter 2, and then we'll transition to Chapter 3, which is metabolism. I asked you kind of look ahead over it, Chapter 3 material, because I mentioned it is a doozy if you don't do your part. It'll just throw you for a loop because it is cell metabolism, which is kind of complex. But before we move to that, let's just review some of the major types of cell junctions which we became familiar with yesterday. In this image that I have shown here, it's looking at two different types of epithelial cells, and here they're joined together with some connective tissue or connective cells as well. And we noted that depending on where the interaction is going to happen, it's going to dictate what type of junction binds those cell types together. So this is the cytoplasm of one cell where my hand is, the cytoplasm of another. And we can see where, well, we didn't cover that exact type, but if you look at this golden junction, it's labeled as a gap junction. What are some characteristics of gap junctions that we learned yesterday? They form a gap, which is kind of like a channel which allows what? Things to go through. And you remember what kind of things are generally transported through gap junctions? Ions. Very good. And what did we conclude about characteristics of ions? They're charged, and so they carry electricity. Okay? Electrical charge or electrical impulses. We'll see this in application in the heart, how the heart all works together, in your smooth muscle in your stomach. Okay, that's how all of these cells work together, is because they are physically connected by gap junctions. And so electricity basically can flow and connect, if you will, the cells together. So those are gap junctions. Another type of junction that we introduced yesterday is called desmosome. We tried to do a physical activity about it. What was the purpose of a desmosome? It binds cells together to distribute tension, to keep cells from tearing apart. So where a cell is likely to be physically under stress, it'd be useful to have a desmosome or some similar type of junction so that it doesn't rip apart. And we also talked about the types of junctions shown here on the top of the image called tight junctions. Those form what type of barrier? A solid, tight barrier. Why? To limit and hopefully prevent the distribution, or the term we'll use, diffusion of substances between cells. So it's a limiting, a binding limiting, diffusion limiting type of barrier. Those are the three primary types of junctions that we'll study this semester or put in an application, but clearly they're not the only types of junctions. There's some variations of those, but those are the ones that we'll use and we'll just point out. What questions do you have about cell structures or extracellular junctions before we move on to our favorite chapter, metabolism? All right, so let's go ahead and jump into the plot, if you will. Metabolism. Cell respiration. Chapter 3. All right, so if we look at Chapter 3 content, it's going to just kind of summarize metabolism. I'll give an introduction here, and we'll review it when we look at the endocrine system, because of our hormones. Some of our hormones help regulate metabolic rate. So we'll become really familiar with it now and then repeat it in the endocrine system. But I'd like to just kind of back up about, well, what the heck is energy needed for in a cell? What do you think? Just list, they don't have to fit into this cute list. What are some things that a cell might need energy for? To move, like what? What could be moved from, like, let's just get some examples. What could be moved from place to place? An entire enzymes, ions. Good. What else? Proteins. Entire cells themselves. sperm cell, etc. So movement, what else might a cell need energy for? Reproduction. Okay, it's an energetic process, so carrying out physical processes that physically involve reproduction. Very good. So you can think of it at a cellular level, or you can think about a person, like an entire female reproducing a baby, another person. What else? What are some other things that a cell needs energy for? Metabolism, which we're going to study. What is metabolism? So digestion is involved, which means what? Processing, getting rid of waste. We could really just, not pulling it out of you today, but we could really just list everything that a cell does. Probably going to need energy in some form to carry out that work. If we were to really just oversimplify why a cell needs energy, we can see it summarized in this simple image. Things like physical work or mechanical work is illustrated like contraction of a skeletal muscle or your heart muscle. So physical contraction or mechanical work. Transporting was also identified. So for example, you did on your handout number 1, active membrane transport. That requires energy to move a molecule against its concentration gradient, so that requires an input of energy. And as you guys also identified using the term metabolism, carrying out chemical reactions. Those also require an input of energy. Okay, so in our body, these are the primary reasons that we need energy. For physical work, transporting substances, and carrying out chemical reactions. So anything that a cell does, probably going to need energy at some point for that process. I'd like to precede that with some basic information that you've kind of maybe taken for granted throughout your science studies, is that energy is always converted or transformed from one state to another. It's going into our laws of what? Which ones? Thermodynamics. Energy is always converted or transformed. It's never what? It's not destroyed and it's never created. It's never created, it's never destroyed, but it's always constant and it's always transformed. And so I tend to inherently, I think I'm just conditioned to misspeak. And I'll say, we make energy. Blah, blah, blah. Is that a true statement? No. What I mean to say is, maybe the cell made ATP. It transformed something into another molecule. And so if I say that energy is made, I'll catch myself and say, I didn't mean that. I meant this molecule was made, or whatever the case may be. And so just to put it in perspective, we know that the way that we understand life on Earth is that the ultimate source of energy does come from the sun, right? Our solar energy, which provides us heat or thermal energy, but it also provides for us light photons. And so we don't have the direct ability to capture those light photons, but we have an indirect method. Okay, so what on Earth can capture those light photons? Things like plants that have the ability to what? Photosynthesize. So plants, bacteria, algae, anything that has light harvesting molecules like chlorophyll, something like that. Okay? Can utilize that light energy and transform it into what type of energy? They would be considered to be phototropic, big plants, the algae or whatever. They can take photomove light, and they can convert the energy, light energy, into what? Chemical energy. Our favorite, most famous chemical that's made by plants is what? The process is photosynthesis. What's the chemical that we like that's made by plants? Well, oxygen, definitely. That's what organelle. Or chloroplast. You heard of sugar? Yeah, glucose, right? Plants make sugars, okay? And so the phototropic organisms can convert that solar energy into chemical energy like sugars and proteins or whatever the case may be. Then we need to do what? To get it. We first need to eat, right? We need to eat. Okay? Any substances which we eat, we can take that and those nutrient molecules and convert them into molecules that we can use for energy transformation. What's happening at each step to the potential energy in the molecules? It's being broken down, if you will. The energy is becoming what in the molecule, the resulting molecule? It's a little bit. It's being broken down, the energy in it is less. Okay? So with each energy transformation, we're going to see energy being released and our byproducts having a little bit lower energy. But they still have what in them? Even though it's lower, there's still energy in it. And so that's what we're going to do is go through these specific chemical reactions. Looking at an efficient, I don't know if efficient is the right word, but a ordered route of energy transformation until molecules get to a point that our cells can use that potential energy. Two terms that we can use briefly to describe metabolism are catabolism and anabolism. Before I look at those definitions, I'd just like to re-summarize or define metabolism. Metabolism is more accurately defined as, right, collectively all the biochemical reactions that happen within, you can look in an organism or within a cell. But if you're looking at a person, you would look at the person's entire metabolism. So it's maybe not what you presumed metabolism is. Okay? So has anybody ever said, my metabolism slowed down since I hit my 30s? Somebody said that? Represent it, right? Your metabolism may or may not have slowed down. You might have been misusing the word. Metabolism is just the total chemical reaction. Maybe you reverse the direction of your reaction. But if we look at all the chemical reactions, they're either catabolic or anabolic. And we've already used the word catabolic, I think, yesterday. What does the word catabolic mean? To break down, if you will. The whole point of a catabolic reaction is to break down why. Why are we breaking substances down? To release energy. It has to do with energy transformation, but if something's catabolized, broken, we're releasing energy. So if that's not in there, yeah, so highlight that. It's releasing energy. Catabolic reactions, yes, they break bonds, but the whole point is to break bonds and release energy. And so we're going to look at some major catabolic reactions for synthesizing a molecule called ATP, which we introduced yesterday, or maybe it was in lab, but we'll become more familiar with today. So we can break bonds, release energy, and what type of reactions? Catabolic, or we could do what? We could build molecules, form bonds, and do what with energy? Use it and store it. Anabolic reactions require energy to make something, but when we make something, we have created what? Maybe. Any molecule is still in your molecule of desire right there. Proteins, ATP, carbohydrates, DNA, or whatever. If I make bonds, I'm storing energy. So anabolic reactions synthesize or make something. I could have used the word anabolic yesterday when we were talking about translation, making what? Translation makes proteins, thank you. So I could have said, well, it's an anabolic reaction. Yes, it is. Or I talked about the activities of the lysosome. What type of reactions occur in the lysosome? What type of reactions would occur in the lysosome? Catabolic. Okay? Because it says we're using enzymes and breaking down the substances. Same thing with lysosome. And so what we're going to do the next 2 days is look at some catabolic reactions to release energy to make this molecule called ATP. So metabolism is just blanketly what? How can we define it? Just metabolism. It's not just breaking down, it's also making, so it's collectively all reactions that are occurring over a period of time. We can subdivide it into catabolic, which implies what? Breaking down molecules to release energy, however it may be, heat or whatever. We'll focus on making what? ATP. You can look at anything to be catabolized. Conversely, anabolic reactions do what? They use to build molecules and store potential energy. Do what? At this point, are there any questions? I'll use those words throughout the semester to remind you we will repeat these terms in the endocrine system. We deal with metabolism again. Figures 3.1a and b help illustrate catabolic versus anabolic reactions. It's not quantifying anything, so we're not looking at a specific molecule, but it's looking at the delta G, or the energy input of a particular reaction. If the energy of the product is less than the reactants, energy is being released, what type of reaction is that? What? It's exothermic, or the word we use today is catabolic, which is an exothermic reaction. Very good. Conversely, if the reactants need energy added to them, let's go ahead and just keep our, what we're adding heat in, it would be considered what? Endothermic. I won't use those in this class. So we're adding heat in, so we're adding energy to it. The products have more potential energy than the reactants, so that would be what type of reaction? Anabolic. This could be amino acids forming peptide bonds to make a protein, whatever the case may be. Right. I'm going to use this image to kind of give you a roadmap of where we're going. You don't have this exact image, but you have a similar image for you on page 2 of your handout. And if you'd like, I can give this image to you. But I just want to kind of give you a roadmap of where we're going. You don't need to draw this down, but maybe make some just cognitive connections today. We're trying to look at energy transformation and metabolism, because we really want to transform energetic molecules efficiently so we can use them to do mechanical work, chemical work, synthesis work, or whatever. Okay? We've got to be able to make ATP. The question is, well, how does it occur? Well, it occurs many ways, and maybe not every way to which ATP can be made has even been characterized. If you're curious, when you go to the lab, next time you go to lab, where I've been sitting, there's a big laminated poster, and it's all the metabolic pathways. And you'll see how intricate and crazy metabolism can be. Once you look at that and put what we're learning today and tomorrow in perspective, it won't be so crazy, but even just today and tomorrow will be a little bit crazy if you're not familiar with cell metabolism. So we're going to try to keep it in a more simplistic view, just an overall view with a little bit of technical detail, and not the level which you would take in biochemistry or say cell biology, something like that. So the whole point is we need to transform energy to make ATP to do work, right? We said in order to do this work, we first need to do what? We need to eat. Do we eat continuously 24 hours? The average person? No. Okay. We have to take a break at least to sleep, right? And go to the bathroom, hopefully take a break to eat, right? So we take in nutrients. We're going to focus strictly on a molecule called glucose. But should we have a diet strictly of glucose? No, don't, right? It'll be bad. But we're going to follow only glucose because that's going to require the greatest amount of processing. It doesn't necessarily release the amount of most ATP, but it has the most processing. Okay? If we eat bloodstream, which makes sense. If you've had a high or low, you're diabetic, right? You know that sugar's in your blood. But we don't keep sugar in the blood just so it can be happy, like on a merry-go-round, right? The sugar doesn't need to stay in the blood. Where does it need to go? To where? Maybe. Okay. It may go to our liver. Why? To store it. So if I eat something and I don't do what with it, I can store it. If I don't use it, I can store it. So one type of nutrients that I eat is, well, they can undergo anabolic reactions to be stored, right? But at some point, I'm going to need to do what with those nutrients? Use them. Or what word could we use? Release, or I could metabolize them, periodic catabolic reactions. So sugars in the blood will eventually go into not just the liver, but pick a cell in your body, brain, skin in your head, wherever, the skeletal muscles. The sugar is going to go into a given cell, where it's going to be metabolized. Sugar has a lot of potential energy in it. If we were to break all of the bonds in each sugar molecule at the same time, what would happen? If I were to just take had something that had the ability to break all of the bonds, making up our, we're catabolizing sugar. So we're breaking it down. So actually in that case it would drop. Okay, so we're taking, do you remember the formula for glucose? Very good. C6H12O6. Let's say we had an enzyme or a team of enzymes that could break down every bond in that molecule at the exact same time. What would happen? Why? We wouldn't be able to use the energy. Why? Well, we want to break it down at some point so we can use it. So if we want to break all of the bonds, if I break a bond, what is released? Energy. And also, what else is released with each chemical reaction? We're breaking glucose. This basic chemistry, when I break a bond, I'm releasing what? What type of energy? A specific type of energy. Yeah. Did you say kinetic? It is kinetic or thermal. Energy is released, or what's released? Heat is another way to say it. If I were to break all the bonds in a single sugar molecule at the same time, I would release a whole heck of a lot of heat, and the body would just burn up, right? Is it a good idea to burn the body, burn the house down in the name of breaking down sugar? No. So if I want to release the energy in glucose and release the heat, what might be an easier way or a better way to release the heat? It's going to have a total amount of heat in it. I don't want to release it all at the same time. I should release it how? What do you say? In the cells, is what we said? In cycles. Very good. In phases. In steps. Release a little now, a little work, release a little bit later. Okay? So I'm still getting the same amount of energy transformation, but it's not all at once. An analogy would be, think about how much heat your house or your apartment needs throughout the winter, right? You don't want to distribute it all on the first day of winter, because by the one you'd sweat, and then, right, or be running out of the house, and then by the end you'd run out. So it makes sense to release a little heat today, a little heat tomorrow, whatever. Same thing with glucose or other nutrient molecules. We don't want to just completely disassemble the molecules in one fell swoop. It's wasteful and counterproductive. A living organism would die. So we'll do it efficiently. And so that's where I'm going is all of these, what to you as a student seem like painstaking, terrible, agonizing steps, is not to antagonize you as a student. It's so that a cell can release the energy slowly, yet, in a sensible fashion to keep the heat reasonable for the cell to maintain what? Homeostasis. So the point is, we're going to take a nutrient molecule focusing on glucose, and we're going to break it down in step 1. We'll continue to catabolize it in another step, and then continue to catabolize it in an ultimate step, so that we can use the nutrient or the heat efficiently or better. Does that make a little bit of sense? The steps that we're going to use include these processes called glycolysis, which you may have heard of before, hopefully, the Krebs cycle, and also what? What's the next thing? What's the next big step? Glycolysis, Krebs cycle, and they're all metabolism. What will be the next big step in metabolism? The electron transport chain. Very good. Those are the main steps that we're going to study. Are these the only ways that ATP can be made? No. It'll be enough for you, I promise. Alright, so we're going to slowly catabolize glucose during the glycolytic pathway, and then we'll get it primed in the mitochondria for the Krebs cycle, which primes the molecules for the electron transport chain. Along the way, we're going to utilize enzymes and coenzymes. We defined already what enzymes are. We know that they're proteins. They're there to help carry forward particular reactions. Whether they be catabolic or anabolic, it doesn't matter. Their job is to carry forward a particular reaction. We also have coenzymes that help specific reactions. And some of the coenzymes that we're going to use over the next couple of days include something called NADH. My notes are not real consistent here, so I have NADH on the first line. Why don't you be consistent with the next one? FAD, put H subscript 2. FADH2. I'll explain that in a moment. And another one called COENZYMES, not real fancy on that. But we're definitely going to talk a whole heck of a lot about a molecule called NADH, and another one called FAD or FADH2, depending on its oxidized or reduced state. And then very briefly about a molecule called acetyl-CoA. How they're going to help these chemical reactions is they're going to carry hydrogen ions, basically. Hydrogen ions can carry what? What are hydrogen ions? They're definitely what? What can they accept and donate really easily? Electrons, okay, which do give us a form of energy transformation. So basically they're going to carry for us hydrogen ions. And why don't you just kind of add a note on there? And electrons. That's what we're going to follow the next couple of days, are hydrogen ions and electrons. I'm not going to have you be accountable for all the individual enzymes in each step of the glycolytic or Krebs cycle or whatever. I want you to understand that these reactions are enzymatically regulated. So each step is going to have its own specific little enzyme carrying forward. If we look at better detail with those coenzymes NAD and FADH, or FADH, excuse me, R, NAD stands for nicotinamide adenine dinucleotide, and you'll see why we abbreviate it, right? NAD. It is partially charged with a partial positive charge, or has a slight positive charge to it. And so what that tells us is it can interact with hydrogen atoms, and it can accept them but it can't accept complete hydrogen ions. It can only take some parts of it. So NAD, nicotinamide adenine dinucleotide, when exposed to two hydrogen atoms, it can take one electron from each and the ion from another. Okay, so let me look at it better this way. NAD can reduce, or what does that mean? Accept electrons from hydrogen. When exposed to two hydrogen atoms, it can take up two electrons, but in that reaction, it releases a hydrogen ion. Just a review for you, NADH would be its accepted electron, so it has been reduced. And in this state, on the left, it would be what? Oxidized. So why don't you go ahead and just review that, write that down. NAD plus is the oxidized form, so it's given up its electron. And in the form of NADH, it's been reduced, which is implied in the hydrogen ion with electrons in there. And to be honest, it might be better if you even made that arrow reversible. So NADH is the reduced form. It's carrying a hydrogen atom and an electron, an extra electron. We'll mention the production of these molecules. We won't use them probably until tomorrow in the electron transport chain. Maybe today we get to this, but I'm not sure that we will. So NAD is a molecule that does what? It sets hydrogen ions, or atoms, to reduce to form NADH. Similarly, FAD, flavin adenine dinucleotide, notice it does not have a charge, so when exposed to two hydrogen atoms, it can pick how much of them? All of them, both of them, to form what? FEH2 subscript 2. So it can pick up the entire hydrogen, both entire hydrogen atoms. So FAD, we would say, as far as reduced or oxidized, FAD is, which one? Oxidized. And FADH is the reduced form. Same thing, you could use make your arrow reversible for the reaction. Energy-wise, which molecule has more potential energy, FAD or FADH2? Which one has more energy, in the oxidized or the reduced form? The reduced form has more potential energy because we just gave it electrons from, or gave two hydrogen atoms basically. So it has more potential energy. So the whole point of these is to transfer what? Energy. The whole point of these is we're just going to transfer energy, or hydrogen ions and electrons. May I help? Oxidized would be the forms where they are not bound to the hydrogen atoms, and once they've bound with the hydrogen atoms, they are reduced. They're reduced, they have more potential energy. Additional questions at this point? Another important molecule we need to address is ATP. What's the proper name for it? The adenosine triphosphate. You've seen something like this when you're studying what? Structure of what? We saw adenosine lining when you're studying our DNA and our RNA, right? You're kind of familiar with it. We've got our little nucleotide, but it has not one, but 3 little phosphates on it, making up its structure. Does anybody know the sugar that's in this molecule? It's not glucose. It's glucose is a hexose, a 6. This one has, what molecule makes up the pentose? Not fructose. It's in a nucleic acid, so we have one of 2 choices. Not dextrose. Deoxyribose or ribose? It's ribose. This molecule is going to have ribose sugar. This is going to have 3 phosphates and adenosine base, making up that structure. So in this image, the phosphates are colored which color? The yellow-red things are the phosphates. You can see 1, 2, 3. This is our ribose sugar, 1, 2, right? Get to the pentose. And this will be our adenosine nucleotide, nitrogen-containing structure. Is that a sign? Yes, and I will address that in just a minute. It is a substrate type. I want to talk about ATP, primary, just basic structure. The root of the molecule is familiar. We've got our base, we have our sugar. But here, I want to look at the phosphates. Phosphates have, are they charged or neutral? They're charged. What charge? Negatively charged. What do we know about charged particles? Likes or opposite? Like charged particles will repel. Oppositely charged particles will attract. Here I have 1, 2, 3 phosphates that are what type of charge? Negative. What do they want to do with one another? Get away from each other, repel from one another. Yet, they're chemically bound together. And that, in a nutshell, is the magic behind ATP. Those 3 negatively charged phosphates bound together, but they have so much, the proper term is electrostatic repulsion. You ever been repulsed by something? These are electrically repulsed. They're bound together, but they don't want to be, right? They want to be separate from one another. So that gives us a source of what? Energy. Those 3, no, it's not 2, it's 3 little negatively charged groups bound together, and they've got a lot of potential energy in them because of the negative nature of the phosphate groups themselves. So what happens is, what we're going to learn is, as soon as ATP is made, what happens to it? As soon as it's made, it's probably going to be what? Used. What can we not do with ATP? We can't store it. Why? Why can't we store ATP? Why can't we store it? You guys just described it a moment ago. Why can't we store ATP? It's very what? Very reactive. So as soon as it's made, it's going to be what? Used. We can't store ATP because it's so reactive. It wants to, as soon as it's formed, it wants to disassociate and release what? Energy. So ATP, we're going to look at how it's manufactured in 3 major steps. Once it's made, it can release a phosphate and release what? Energy. So look at the simple, just an oversimplified view of ATP. If we were to catabolize it, one phosphate at a time. If I release a terminal phosphate, we said we're releasing energy. The rest of the molecule remaining is what? ADP. Adenosine what? Diphosphate. So the ATP molecule has released a phosphate. We call it ADP. It's still reactive, but a little bit less reactive than ATP. So we still have energy that we can transform, just not as much. And yes, when ATP liberates, if you will, its terminal phosphate, the terminal phosphate goes to something. The something gets what? Gets what? I'm giving it the little reactive phosphate. I'm giving it what? Energy. It gets energized, but that terminal phosphate, it's just the phosphorus and the oxygen atoms. There's no carbon, so we call it inorganic phosphate. That's what the I represents. The subscript I stands for inorganic phosphate, or I'll inaccurately say just plain old phosphate transferred out. And so if we split ATP, yes, ADP is going to remain. Inorganic phosphate is released, and energy is transferred to wherever that phosphate goes. What questions at this point do you have about the kind of basic structure of ATP? That's how many phosphates? 3. They have what kind of charge? Negative. They're bound together, so they want to repel. And then when ATP transfers off its terminal phosphate, the thing that it transfers to gets energized. It is a hydrolysis reaction, so if you want to include the water on there, that's fine. If not, it's okay for the kind of overview in that. We're going to look at 3 major steps, as we identified earlier, not the only steps for making ATP. The steps which we're going to study for ATP synthesis include a process called glycolysis. Lysis means what? Breakdown. So what type of reaction? Catabolic reaction of sugars. We're going to split our sugars, but not 100%, so we need to do what to it? Well, before we could store it, but we're going catabolically, so we can continue to break it down or catabolize it. The next major step is the citric acid cycle, or the what? Krebs cycle. We may call it the tricarboxylic acid cycle. And then after that, you identified as well the electron transport chain. So these are the major steps that we're going to study between today and tomorrow. I'm going to come back to this list, and we're going to use some new terms that I'm going to introduce to you next. All right, so the question is, well, if I'm moving phosphates, moving energy, is there a better way to state that? And yes, there's a more concise way to state that. And the term is called phosphorylation. Phosphorylation. I'm going to focus on this word first. Phosphorylation means a molecule has gained a phosphate. Just looking at the word phosphate, phosphorylation, not the prefix yet. Phosphorylation means something has gained a phosphate and really has gained what? Energy. So phosphorylation means something has gained a phosphate. We've changed its structure, we've changed its function. So in phosphorylation, we're doing what to a molecule? Very good. In phosphorylation, a molecule gains a phosphate, which implies, right, energy. We've also changed its structure, we've also changed its function, all all kinds of good stuff by just plunking on, if you will, a phosphate group. If something gains a phosphate, something must have lost a phosphate. There's a word for that. What do you think a molecule that's lost a phosphate? Dephosphorylation. So a molecule can be phosphorylated, or it can be that means it has what? And lost what? And we've also changed its structure, and we've also changed its function. Make notes in here. Okay. So if I use the words phosphorylation and dephosphorylation, are you okay if I use those words? Because we're going to add to the complexity. The question is, well, how is a molecule phosphorylated? How is it? How does this happen? And there are two major ways in which a molecule could be phosphorylated. The first is described as called substrate level phosphorylation. In other words, a phosphate is transferred from one substrate to another substrate. What does that word substrate mean? Generic word for any molecule, right, compound, whatever. A good visual way to illustrate, are there any football fans here? Okay, it's basketball season, right? Basketball fans, right? Okay. Think of your favorite sporting event, okay, where a athletic ball is transferred from one person to another. The athletic ball, the football or the basketball, will be the phosphate group. Okay? Let's go with football. I'm more familiar with that. Okay. If I'm the okay, I'm really not familiar with it, but I know the quarterback does. Okay, so I'm the quarterback. Okay, you guys probably know it. So the snapper, right? Is that the person who's in the middle and hands the ball back? Who's that person? The center? The center? Okay. The center. He is phosphorylated, right, at the start of the play, right? He wants to phosphorylate the quarterback. So he gets phosphorylated, and he reaches back and alright, can you be my thesis?
Yeah.
I'm phosphorylated, and I'm going to become dephosphorylated, and now she is phosphorylated. Can you see how I directly transferred the phosphate group from me to her?
So you lost your energy then
I've lost a little bit of energy, but I'm not useless. I could become more useless. I could be rephosphorylated, and I could phosphorylate you. Yeah, I could phosphorylate you, and you could I hope you don't mind there. I'm talking about germaphobe. Okay, and now you can phosphorylate me, and I can phosphorylate her back.
So you can just go back and forth.
Yeah. We were just transferring what? Energy. Phosphate group. When it's in a more simplistic fashion, that just direct, hand it off, it's called substrate level phosphorylation. A phosphate is on one molecule, enzymes will transfer it over to another. So here I have an example of like molecule X. Name your molecule, whatever. It has a phosphate. It can directly phosphorylate ADP or any other molecule. And now ADP was phosphorylated to form ATP. And molecule X has now become dephosphorylated. It's not useless. Okay, we could reverse it. But the key is, in substrate level phosphorylation, the phosphate is just transferred. It's directly transferred. There's nothing real trickery about it. It's just a direct transfer from from the center to the quarterback, from glucose to whatever, okay, fill in the molecule. The direct transfer. If there's no trickery about it, if you will, and it's kind of simple, there must be a way that's a little bit more what? Complicated. But with complication in a biological system, what do you think the advantage is to going around a more difficult path? It's what? It's more abundant. Okay? And so the other type of phosphorylation that isn't direct or substrate level is called oxidative phosphorylation. Oxidative phosphorylation is still going to phosphorylate a molecule, but we're now going to have to transfer what? Oxidative, what? Kind of, because they're carrying oxidative reactions transferring electrons. Electrons. We're going to move about electrons and do some work. Oxidative phosphorylation is going to require some oxidative reactions. And this image is really simple. It doesn't really illustrate the complexity to it. But we're going to have the ability to transfer inorganic phosphate, something that's not bound to a molecule, over to ATP. So it's almost like the football sitting on the field, and somehow electrical forces put it in the quarterback hand. Another person in the substrate level phosphorylate the quarterback. Something, I don't have a real tangible example in football, but something extra lifts that football and puts it in the quarterback hand. And then the quarterback can phosphorylate the hand of football. And so, I really can't think of anything real in athletics that would kind of equate to electron transport. So maybe wind. The wind would gust and gust and gust and gust and gust until the football gets high enough to fall into the quarterback. Kinda. You can do like a kickoff return. So, yeah, that would be a good one. So the football is in the air. It did originate from somewhere. But in the air, it's not attached to anything. And then the receiver is phosphorylated. That's a great example. But along the way, a bunch of other stuff. So it makes it a lot more complex. But visually, that's a great example. Thank you. All right. So in oxidative phosphorylation, we said there's going to be a lot more steps, but what? More abundant energy will be produced. More ATP molecules will be released. So let's go ahead and add this. Substrate level is direct. If there's one phosphate transferred, how much ATP are we making? One. In oxidative phosphorylation, we may make a couple dozen ATP molecules with a single round of punt return. So I guess that would be an example. Maybe one person kicks a football and 20 fall from the sky. And there's 20 receivers. That would be a good example. And how many touchdowns can we get? Up to 20. That would probably help illustrate a little bit better. So it's more abundant, but it's going to take a whole lot more what? What'd you say? A lot more work. Okay, but the payoff is good. And so let's go back and use these terms for those 3 steps that we're going to study. Glycolysis is a quick reaction. What type of phosphorylation do you think we'll have? Substrate level phosphorylation. So on that list that I said we're going to come back to, I want you to acknowledge that glycolysis produces ATP by substrate level phosphorylation. Substrate level phosphorylation. The correct cycle the electron transport chain transfer the electrons must be oxidative phosphorylation. The electron transport chain is oxidative phosphorylation. We're going to use electrons to make a whole heck of a lot of what? ATP. So a molecule can be phosphorylated, gain a what? Phosphate, and gain energy. A molecule could likewise be dephosphorylated and lose a little bit of energy. It's not useless, right? It can still be used. That phosphate can be transferred directly by what type of phosphorylation? Substrate level, or kind of indirectly by oxidative phosphorylation. Let's now look at that next image on your handout. This is a metabolism kind of oversimplification, but I want to point out some things here. Kind of like a metabolic simplified roadmap. And you can kind of see three pathways in the colored image. We have the yellow proteins, the green carbohydrates, and then our lipids, fats, and the purple pathway. This is illustrating what I tried to mention a moment ago is we don't need just sugar for energy. We need a well-balanced diet of proteins and fats as well. Because each of those molecules can be metabolized to release what? ATP and other intermediates along the way. We're going to follow this narrow pathway for the catabolism of glucose. So if you scrutinize this image a little bit, you notice the first processing of glucose, of any sugar really, occurs more than processing of our other nutrients. Implied by the kind of the arrows, right? If I want to catabolize glucose, I need to first break it down during the glycolytic pathway, form something called pyruvic acid, then go on to the Krebs cycle and the electron transport chain. Contrasting that to our lipid catabolism, it can be catabolized in jumping in the pathway, if you will, later compared to sugar. You notice how it's kind of entering in the intermediate step prior to the Krebs cycle. So fats can enter the same pathway but later. Additionally, proteins can be catabolized and enter the pathway later. The point is, if you understand basically how sugars are broken down, you can then appreciate how proteins or lipids can enter into the catabolic pathway, the Krebs cycle or wherever it may be. So that's why a lot of your classes focus on sugar. If you have an idea about sugar, you have an idea about the catabolism of our other nutrients. Obviously, some nutrients have more bonds, like our fats have more bonds and they have more, what in them? Energy storage and releasing ability. But it's going to take more processing for our sugars. Okay? And so that's why we're going to follow this little narrow pathway. Glucose, as I've been leading up to, is clearly going to be an important molecule, right? For have the word fuel, you could say nutrients or energy, okay, for most organisms. And the key thing is it's oxidized, or what does that mean? Oxidized, we're taking off what? Electrons. We're releasing electrons. Circle it. How? Slowly. Why? Why do we want to do this not fast? We don't release the heat so fast, do we? We want to do it more slowly so we can be what? Maintain homeostasis, we want to be efficient. So we're going to take off electrons, we're going to take off energy slowly from our sugar molecules so that we can make ATP in a sensible fashion for the cell to maintain homeostasis without raising our temperature. In this pathway that we're going to study, the first step that we'll start with is glycolysis. And glycolysis does occur under anaerobic conditions. What does that mean? Without oxygen. I want you to restate your comprehension of a metabolic reaction that's anabolic, I want you to look, it does not, what? Use oxygen, but hopefully oxygen is what? Hopefully it's present. So even though oxygen is present, it's just not what in this pathway? It's not used in this pathway to do what? To do what? What is that representation? Electrons. Oxygen does not accept electrons in this pathway. Just because glycolysis occurs doesn't mean that you're choking or suffocating or whatever. Hopefully you're respiring normally. Hopefully your blood oxygen levels are still at set point levels. It's just not used here. It's used elsewhere. Very which process? Which one? Not in glycolysis. We're going to go glycolysis is anaerobic, so we're not using it here. Where are we going to use oxygen to pick up electrons? The electron transport chain. So that'll be our aerobic pathway. So glycolysis is anaerobic. It doesn't do what with oxygen? It doesn't use it, but it has no implication of its presence or absence. Does that make sense? Hopefully oxygen is present always inside your body. So glycolysis is certainly going to occur under anaerobic conditions. So the electrons are going somewhere besides oxygen, molecular oxygen. The glycolytic pathway, or you can call it glycolysis, does occur in 10 separate reactions in the cytosol. This is important where it occurs. It occurs in the cell cytoplasm or cytosol, and it's enzymatically regulated. It's important to understand it's not in the mitochondria. So we can make ATP certainly in the cytosol very easily during glycolysis. And we identified as the anaerobic breakdown, or I could say catabolism of glucose. And the final products of the glycolytic pathway include two. You can call it pyruvate, or you can call it pyruvic acid molecules. Pyruvic acid is pyruvate. It's just an alternative name for the same thing. So it produces the glycolytic pathway will split glucose into two pyruvic acid molecules. And along the way, it'll release 2 molecules of ATP. We don't have anything to compare it to at this point, but 2 isn't very much ATP. But the point of glycolysis is not really to make a lot of ATP, but to do what to glucose? Break it down slowly, right? In respect, right? We're going to do it efficiently. So the point of glycolysis is to break glucose down into pyruvic acid, which still has what in it? Energy. Still has more chemical bonds that can be broken. In the glycolytic pathway, it's really interesting because you're like, well, we're trying to make ATP, right? That's where we're going. And it's interesting, in the glycolytic pathway, it actually spins ATP before it can actually release energy. ATP. So glycolysis is kind of expensive, if you will. It takes 2 ATP molecules to get it going. If we end up with a net production of 2, really how much ATP is totally made during glycolysis? How much? 4. 4 are made. If I spin 2, I'm in balance. Think about your bank account. If you have a balance of 0, and then you spin 2, your balance is negative 2. And if I want to get up to positive 2, I need to add in first 2 to bring me back to 0, and then another 2. So I would have to put how many dollars in our bank account to get $2? 4. Glycolysis kind of does the same type of spinning, if you will. It spins 2 molecules of ATP to get glucose ready for catabolism, and then it releases a round of 2 and then another round of 2 molecules of ATP. But how many usable amounts of ATP are made Really only 2 molecules of ATP, net molecules of ATP, are synthesized. So when you go into, somebody been in a biochemistry yet? So something like this will look really familiar to you. I'm not going to do biochemistry to you, right? I'm not going to do that to you. But I keep mentioning cell biology. Anybody taking cell with me this fall? Nobody? Come on. We'll get to study this in great detail. Each step, each transformation uses a specific enzyme to form a specific product. Okay. And if we look at starting at the beginning, starting with glucose, and in the end we end up with pyruvic acid. Pyruvic acid, you can still see, has bonds in it, which we know we've been simplifying with energy in it, which can be broken down later. But we can also see the release of ATP. And I've been forgetting about something really important. What else is released in ADH? Is this overall reaction in your handout? Yes. Okay. On the byproducts, on the right side of the arrow, we know, make a note under about the pyruvate. It has more what? It has more potential energy. ATP can be what? What can it be done with? We better do what with it? We better use it. For whatever, chemical, mechanical, whatever that cell needs it for. NADH has what? It has more energy. The hydrogen ions also have what in them? Energy. And water is also a byproduct which can be used for whatever the cell needs. And so what we're going to do tomorrow is follow pyruvic acid, NADH, and hydrogen ions, see how we can extrapolate their energy to make even more what? In which molecule? ATP. Very specifically, we want to make ATP. Over the weekend, over tonight, homework number 2 will help you a ton. I would go in, has anybody done it yet? Go do homework number 2, at least watch the videos. If you don't want to answer the questions, at least watch the videos tonight so that you're not petrified tomorrow. Okay? I'll see you in lab or in class tomorrow morning. If you're late, you'll want to come pick out handout number 2 and submit handout number 1.